Good morning, good afternoon, and good evening to everyone joining us from wherever you are around the world. Distinguished guests, ladies and gentlemen, it is my great honor to welcome you all to this highly anticipated awareness seminar. Before we begin with our main presentation, allow me to provide a brief overview of the initiative that brings us together today. Install Africa, Innovation, Science and Technology for Action and Learning in Africa is the result of collaborative efforts initiated in October 2023. This initiative leverages the Drosophila melanogaster fly model and state-of-the-art infrastructure to enhance fundamental research capacity in Rwanda. Rwanda has made remarkable strides in advancing its scientific capabilities, recognizing the pivotal role of research in sustainable development and public health. DrosAfrica was born from a collaboration between the Foresight Institute of Research and Translation Rwanda, or FIRAT Rwanda, and its technical partner, DrosAfrica UK, in conjunction with key national institutions such as the National Council for Science and Technology, Rwanda Biomedical Center, the National Industrial Research and Development Agency, the University of Rwanda, and the Rwanda Food and Drug Authority. This collaboration embarks on a 5-year comprehensive capacity development plan aimed at cultivating the next generation of scientists and fostering research excellence across medical, applied, and fundamental sciences. From healthcare innovation to agricultural production, this partnership envisions driving progress in public health and sustainable development across Rwanda and beyond. It is against this background that we convene today, united in our shared commitment to advancing science, technology, and public health for the benefit of our communities. With that foundation laid, it is now my distinct pleasure to introduce our invited guest speaker.
It is with great honor and pleasure that we welcome Professor Rose Hayeshi to join us today for our webinar, Growing a Preclinical Platform, Lessons from the Ground Up. Professor Hayeshi is the director of the Preclinical Drug Development Platform, or PCCDDP, at the Faculty of Health Sciences, Northwest University, South Africa. She is also a Calestus Juma Science Leadership Fellow through the Gates Foundation. Professor Hayeshi holds a PhD in biochemistry and has co-authored more than 30 scientific publications and 3 book chapters. She has also trained a number of postgraduate students and postdoctoral fellows, contributing to the development and mentorship of emerging researchers. Her research interests are centered on human drug metabolism and pharmacokinetics, with a particular focus on humanized mouse models for preclinical drug development. In the area of preclinical development, Professor Hayeshi works within interdisciplinary and inter-institutional collaborative teams across different aspects of drug and vaccine development. She leads key preclinical drug development studies and is also championing initiatives aimed at enhancing capacity for translational research and innovation in Africa. Her contribution to science extends beyond her research activities. Professor Hayeshi actively participates in both national and international scientific communities, serving on various committees, engaging with professional societies, and collaborating with researchers across multiple countries. Her experience in leading a preclinical drug development platform makes her especially well-placed to speak to today's theme, not simply the science of preclinical development, but the experience of building and growing a platform within a research environment. And that is what makes today's conversation particularly valuable. As we explore growing a preclinical platform, lessons from the ground up, we have an opportunity to hear directly from someone with experience in this space and to learn from the work, collaborations, and perspectives that have shaped her journey. We are therefore delighted to have Professor Hayeshi with us today, and we look forward to hearing about her experience, the lessons learned along the way, and her perspectives on building capacity for preclinical research and translational innovation in Africa. Ladies and gentlemen, please join me in warmly welcoming Professor Rose Hayeshi. Professor Hayeshi, the floor is yours.
Hey, thank you very much for that wonderful introduction, and thank you to the organizers of the Install Africa webinar series for inviting me to present here. Good day to everyone that has joined us, hopefully from different parts of the continent. and other parts of the world. So let me just share my slides here. I'm going to take you on a journey that was taken by the Preclinical Drug Development Platform about how the lessons we've learned in growing the platform. I'm based at the Faculty of Health Sciences, Northwest University in South Africa. So just a little bit of geography to show you where we are based. The Northwest University has 3 campuses. So I am on the Potchefstroom campus, which is here, not very far from Johannesburg, for those who have been to South Africa. And then we have another campus called the Mahikeng campus. This was here, closer to Botswana. And then we have a Vanderbijlpark campus also close to Johannesburg. So that is where we are based, just to give you an idea. And a brief overview. So the Northwest University is also highly research focused, with the mission to benefit society through provision of knowledge, excelling in innovative teaching and cutting-edge research, and importantly, also focused engagement with the community. And our principal and vice chancellor is Prof. Bismarck Jobeka, as you will see there. So we have 8 faculties, and my department is in the Faculty of Health Sciences. Let me just find my pointer here. So we are in the Faculty of Health Sciences. And since this webinar series is about contributing to strengthening research capacity, just to show you a little bit about how our university views that. So we are in the top 4% of the world's best universities. And we have quite a lot of growth in our research outputs. As you can see here, for example, journal articles steadily increasing from 2020 to 2024, and also our postgraduate students. So as I've mentioned, we are in the Faculty of Health Sciences. The faculty is led by an executive dean, and under the dean, we have a deputy dean for research, innovation, and postgraduate studies and community engagement, deputy dean for teaching and learning, and then a deputy dean for stakeholder relations. Then we have this group of departments here, of which the PCDDP is one. We report directly to the executive dean. This is because within the faculty, we are part of 3 other facilities where our focus is more commercial and service delivery based. We do have students, but this is where we lie in this specialty as a platform, as a national platform. So how did we get here to be in this position? I'll just give you an idea of the journey to the platform. So we start with the first lesson, that there was a need that needed to be filled. And I will start by explaining that need, by explaining what is a drug development pipeline, how are medicines developed. So if you look at the drug development pipeline, there are 2 areas, which is drug discovery. This is the process where scientists are looking for new molecules that could someday lead to becoming drugs. And then in drug discovery, so you have the basic research and drug discovery going on. And then you have the second phase, which is the drug development phase. And this is where the rigorous testing of new drug candidates for safety and efficacy is done, so that we can eventually end up with a drug, an approved drug in the market. And preclinical drug development is the bridge between discovery and development. So this is where you are doing the testing, could be in tubes, in test tubes, so to speak, and in animals, which is what we do in terms of getting the safety and efficacy data before you can advance these potential drugs, new drugs, to testing in humans. So if you look at the drug development value chain in South Africa, and I'm sure this is common for other African countries as well, there's a lot of expertise in the discovery phase. And with all our natural biodiversity, we also have a lot of drug discovery researchers identifying potential drugs from plants, for example, right? So we know there's a lot of activity there. And also clinical trial expertise is quite well established as well in South Africa, at least it is. So what is then the reason that we actually don't have a drug that has come out completely developed on the continent, right? And South Africa determined that this is one of the gaps is the preclinical development gap. So they focused on this, covering this gap with the aim of strengthening the pipeline so that in the future we can have a drug that's developed end-to-end within South Africa. And that is how the Preclinical Drug Development Platform was born. So it was a joint initiative between the South African government of Department of Science, Technology and Innovation and the Northwest University. It was established in 2011 as a national facility supporting innovation of pharmaceutical development, focusing on preclinical drug development. And as we are hosted in a university, we also teach postgraduates, and while supplying expertise and innovation to scientists and businesses locally and internationally. So our vision is actually to meet the stringent regulatory requirements, comply with international pharmaceutical best practices, and to deliver a world-class service in preclinical drug evaluation. So we are at this critical bridging point. And similarly with vaccines, although our name focuses on drugs, but vaccines are also part of therapeutics. And with the advent of COVID and the realization that Africa needs to develop its own vaccines, we've also have more activity within the vaccine development pipeline consortia. So within vaccines, the preclinical testing will be focusing on immunogenicity testing and safety toxicology testing, which you will hear a little bit more about later on. So that is where we are playing. And just to show you what the progression was, Prof. Anne Krobler, she was the former director and founder of the PCDDP. And I joined here at the end of 2014. So this is where I joined the platform. The university had an existing animal care center. And when they won the bid to host this platform, they got funding from the government. So this is 33 million is, what is it in dollars? It's about 1.5, I think, $million or so. And then the university matched it with half. So Prof. Krobler and the team went to visit other labs in Europe to benchmark, and came back and they redesigned the whole building. And since then, when it became operational, they had a number of projects with universities and other institutions within South Africa. They had students coming out. They did work for industry over 100 million rand worth. That's about $5 million, acquired specialized equipment, and then also got accreditation and validation, which I will tell you a little bit more about later on. So this is just in a nutshell what happened in the early years. Then the second lesson is that the people are the platform. You cannot have a building and not have people operating it. So I will start with the people that we have working towards this vision. So that is our team. You can see it's a very small team. And this is how we are structured. Some of this will become a bit clearer to you later on because it's linked to our accreditation, GLP compliance certification, rather. So I am the director, and then with my GLP hat, I'm also a test facility manager, which I'll explain to you later on. And we have 3 other academics. So we have a senior lecturer and 2 associate professors, and they also double up. One is a facility coordinator, another backup quality assurance, and one is a study director. We have a dedicated quality assurance manager. Quality is important to us. And then we have an animal facility, and for that, this is the team that leads the animals. So this is the team as we have it now. And we have scientists, quality assurance, we have archive capabilities for our GLP function. So this is part of the team. And we also have expertise in laboratory animal handling. So we have our veterinarian and vet nurse and a number of lab animal technologists. Today is actually the first day of one of our new LATs, Mr. Clifford Maine, so I don't have a photo of him. But if you look at our lab animal technologists, they have combined over 80 years experience to do the work that they do with the animals, which is, as I said, is important, and we couldn't do the work without them. So now to the ecosystem. What is it that is needed to deliver the science, right? We have the people, what else do we need? And bringing you back then to the roadmap. So I'm going to just remind you that we went and designed a whole new building. I'll show you what that building looks like. We've done studies with animals. I'll tell you about the animals and some of our instrumentation, and also moving on from there, it will be about the accreditation. So we have a really state-of-the-art infrastructure. It's environmentally controlled with building management system, data management systems. We have pressure cascades running through. I'll show you pictures just now. We have HVAC, so all the air is controlled, and the temperature and humidity. And at any given time, you can see on a screen what the temperature, humidity, and pressure is in any given room where we house the animals. And we have a basement level floor where we breed. We do a lot of breeding of animals there, and that is specific pathogen free. So it's a very controlled area. It's not sterile, but it's a controlled area giving certain quality of animals. And I see here, I probably haven't mentioned, but the type of animals we have is mice and rats, which I'll tell you about in a few minutes. But just to then show you the facility, we do have a virtual tour, which I will also show at the end of the presentation. But for now, I will show you just the pictures to see what it looks like. So this is our entrance area, and you can see here we have this red line. So beyond this, you have to put on certain PPE. We have sensors in the building. So if you look on the roof, you will see these are the ones that keep track of what is going on with the temperature, humidity, and pressure within the building. And that feeds back to the building management system, which then ensures that everything stays within spec. We have night and day cycle reversing. So within a study room, if you want, we work with rodents, as I said. So if you want to work, and rodents are active during the night. So if you want to work with them during their active phase and you don't want to come in at night, then you can reverse the light so that within the study room it will be dark during the day, and then they think it's nighttime. And we have this red film on the doors, which prevents light from the outside coming into the room. So when the light from the corridor comes into the room, it's red light, which means we can see, but the rodents cannot see through red light. So to them, it's still nighttime and they'll be active. So one of the other lessons is understanding the behavior of the species of animal that you're working with. Our animals are housed in what are called individually ventilated cages, and each cage has a blower attached to it, the cages are put in racks, and each rack has a blower attached to it, and these blowers are the ones that control air supply to the cages. So each cage has an inlet and outlet pipe, so you can see the cages are closed, so each cage gets its own supply, air coming in and going out. And that would mean if the animals in this cage are sick, for example, they will not affect animals in other cages. We also have metabolic cages if you want to do studies to, for example, determine if you're looking at drug metabolism, for example, when you've given the drug orally to the animal, you can then separate the feces and the urine to determine the biodistribution of your drug of interest. Of course, you can also use them for nutrition studies. And this is our theater where we can do surgical procedures on the animals. And when you euthanize them, we also do it in the theater. We have cryopreservation and -80 and liquid nitrogen as well for storage of cells and samples. And we also have specialized cage washers. As you saw, it's a lot of cages. So these are automatic cage washing machines that we use for cleaning up. And then we have pass-through autoclaves and hatches to go from the dirty side or into the SPF area. So when they've washed cages, then there's a pass-through autoclave, which means then they get clean cages and food and bedding into the SPF area. We also have a cell culture lab for part of one of the cancer models that we work with, where we grow cancer cells and then we can put them into the animals, inject them in the animals to induce cancer. And then in terms of infection, which we know is one of the important areas of research, of course, on the continent, we have this container lab. It was biosafety level 3, but now it's been downgraded to a biosafety level 2. So we're currently fundraising for biosafety level 3 modular lab that would have animal housing, PCR, as well as cell culture. But this current lab is just small enough for 2 people to fit inside. And currently, for example, there's my postdoc is doing a project in there with malaria infection. So our rodent models, we have mice and rats. The housing capacity is over 2,000 mice and over 4,000 rats. We have standard laboratory mice and rats listed there. We also have specialized models. So, for example, immunodeficient models that we use for cancer studies. So this is a nude mouse with a tumor growing. So what we did is we injected cancer cells under the skin, and then that grows into a tumor, and you can use that to study your anti-cancer drugs. And we have other specialized immunodeficient models. We also have a model for studying mycobacterium tuberculosis infection, and then other researchers within the faculty who do neuropharmacology work have their own specialized rodent models. And for my personal research, I have humanized models for drug metabolism. Humanized means that they are transgenic, so they've been genetically modified to express certain human enzymes. So sorry, this is just a closer look at what the individual ventilated cage looks like. In terms of the studies and instrumentation that we have, so our unique selling point is that we can do acute and chronic toxicity studies according to OECD Good Laboratory Practice guidelines, which I'll explain later on. I've mentioned drug metabolism and pharmacokinetics. We can do efficacy. So we've done cancer, diabetes, and malaria. We are not really, that's not our main focus point. However, should someone require a specialized disease model that we do not have, we're very happy to work with them to develop the model so they can do their work. We also have ethnopharmacology research. So one of my team members is working on, actually 2 of them are working on ethnopharmacology. And one is looking at use of plant extracts for looking at using it for depression and similar diseases. And then the other colleague is looking at herb-drug interactions. So he, for example, is looking at the effect of inhalation. So if you have medicinal smokes, how does that interact with the drugs? And with that, we have a specialized instrument where you attach your animals to these holders here, and then you can then pump or expose rather the mice or the rats to inhalation. So you can use that to study medicinal smokes or even just smoking in general. This is our new liquid chromatography mass spec that's newly acquired. It's even less than a month old. And then we have a specialized ELISA plate reader as well. So one of the things we've learned is that obviously we have unique risks in low resource settings, and one of those is stable electricity for sure. And I'm sure this is relevant for a lot of us. In South Africa, there's something called load shedding, where they, when the electricity supply is low, then they cut off power and so on. So we had to ensure that we have mitigations for stable electricity. So the facility has its own backup generator. And recently, we've just also installed backup solar power to reduce dips, because every time the power goes off, and there's always a lag between that and the generator coming back on. And then we have a lot of sensitive equipment. And even though you have your backup power, these energy dips or power dips end up affecting the sensitive equipment. Of course, it's also high maintenance costs. Water quality is not always the best, so you need water filters. And then, of course, I think supply chain and logistics. In terms of importing things, we have to import our animals. we do breed our animals, but every now and again you need to replenish your stock, and sometimes just getting the import permit for your animals can take quite some time. So these, I'm sure, are common challenges that we all face. The next lesson we learned is that, I'd like to share, is that quality builds trust. So we are very big on quality, and we have 2 key certifications and accreditation. So we are compliant for Good Laboratory Practice, OECD, since the past 10 years, and ALAC as well, since 2017. ALAC is an international accreditation system for the care and use of your animals. So it's a private nonprofit organization that promotes humane and ethical treatment of animals used in science. I took this from the website, and it safeguards quality animal care and scientific validity. We all know that happy animals means not stressed animals, which means you get good data. In Africa, there are only 3 countries that have ALAC accredited research animal facilities. So in South Africa, it's just us. In Mauritius, there are 4 facilities, and then in Egypt, there are 3 facilities. So it's quite a prestigious accreditation to have. We are inspected, I think, every 3 years. They actually come in person and they inspect. They inspect the way we house the animals, the way our SOPs, and they also inspect the ethics committee. The Animal Research Ethics Committee is part of this accreditation. And also, they also very much, they also incorporate the occupational health and safety of the team working with the animals. So that's the ALAC. And then coming to the GLP, as I've said, if you look at the drug development pipeline, the preclinical studies are bridging in the middle, and there is where you determine safety and efficacy. The safety is important because that data is what is used to inform clinical trials. So before you go into humans, you have to ensure that your drug of interest is safe. And such studies, pivotal toxicity studies, are conducted according to strict regulatory guidelines. That's the OECD GLP guidelines. GLP is a quality system concerned with the organizational process and the conditions under which non-clinical or preclinical health and environmental safety studies are planned, performed, monitored, recorded, archived, and reported. So it's amenable, it's similar to GCP for those who are familiar with clinical trials. And for those who are familiar with manufacturing, it's similar to good manufacturing practice. And in South Africa, the SANAS, which is the South African National Accreditation System, is the one that's responsible for inspecting facilities for certifying them GLP compliant. So we get inspected by SANAS every year or every 2 years. The number of tox studies we've conducted over the past about 10 or so years is a lot of oral toxicity. We've done acute mostly and a few repeat dose. And our customers are normally universities within the continent and also consortiums, normally South Africa. So there's a lot of South Africa based here, and part of me also presenting to install at this webinar is to increase the use of our facilities, sensitize the rest of the continent that we are in existence. I would be really, really happy to work with you. And then other studies, as I've said, we do efficacy. So for example, we've done phytochemicals in diabetes. We do a lot of cancer xenograft studies, a few vaccine work here and there, and even microbicides. So we are quite diverse in the type of work that we can do. Yes, so as I mentioned, partnerships. To us, partnerships matter. All of this is towards developing new drugs, so it's towards global health, and we cannot do it in partnership. And as you see, because preclinical drug development sits in the center of the product development pipeline, we cannot do this without partnerships. Within South Africa, we have, of course, academic research collaborations, product development consortia, which for me works really well. So, for example, we're part of the South Africa Messenger RNA Vaccine Consortium, which has a number of, built out the whole pipeline from the people who discover and design vaccines all the way to manufacturing and be a part of that. Other local consortia like the NRF Pandemic Institute that is still in planning. And of course, as I said, we also then do service delivery, so we can work as in collaboration, or people then pay us to do the work for them. Internationally as well, of course, we've got academic collaborations, we can do student exchange. And once again, importantly, we're part of research and product development networks, such as the Grand Challenges Africa Drug Discovery Accelerator. We are part of the CEPI Preclinical Models Network. So CEPI is the Coalition for Epidemic Preparedness Innovation, and they are focusing on vaccine development. So we are part of that network of labs as well. Whenever they need, one of their partners needs preclinical work, then they come to us. So we've joined CEPI just about 2 years ago, and we'll be performing the first study for them soon. We're also part of Spark Africa, which is looking into product development, capacity building on the continent. And we're also part of the International Mouse Phenotyping Consortium. The IMPC is a consortium that has, when the mouse genome was done, they knock out every single gene in the mouse to see what is the effect on the mouse, and then they can kind of extrapolate that to humans. So since about 2018 up to current, that's when a lot of our networks got built. So we joined the IMPC in 2018. 2021, after COVID, we were part of a local group of consortium within South Africa with one university and ourselves, and we were the lab. We had a researcher developing, not ourselves, but there was a researcher developing a COVID vaccine, and we did the animal trial for that, and that came out in the news. We've been part of an EU program for training students, and we're also part of a local product development consortium within South Africa. And from 2023, that's when we joined Spark Africa and the South Africa Messenger RNA Vaccine Consortium that I mentioned. We are part of the DMPK Africa Network, which is part of the Grand Challenges Africa Drug Discovery Accelerator. And in about 23, 24, we were part of Matrix, which was a consortium, global consortium. Well, not global, but yeah, there are a lot of US labs, obviously it's USAID, and a number of institutions in South Africa, Kenya, and Zimbabwe in terms of advancing R&D of innovative HIV prevention products for women. Obviously, unfortunately, when the funding got cut, then that project did not continue. And then in 2024, we held our first IMPC spring meeting on the continent. So we brought all these IMPC researchers to Africa. And I think we all know, in terms of looking at the models that we have available to us, the genetic engineering aspect is not always well developed. So because through a membership with IMPC, it means we have access to working with researchers who can make genetically modified mouse models. For example, for if somebody wants a specific disease or like a rare disease, then we can work with the IMPC to see how we can develop that. And then, as I said, with part of CEPI, and then 2025, we are then launched ISSX Africa. So this is the International Society for the Study of Xenobiotics. Is a long-standing, since the 80s, international society for drug metabolism and pharmacokinetics. And in 2025, we then launched ISSX Africa, myself and other researchers on the continent. So I just want to tell you a little bit about the Preclinical GLP Toxicology Hub, because one of the things that we learned is just putting an animal facility, that's not the end all and be all. The preclinical stage itself is a whole ecosystem in itself, right? And this came out strongly when we had to do a study during COVID, where it was realized that there are still some capacity gaps, right? So the product developer sends us the product for testing, and it must still go out for histopathology. It must still go out to other labs for hematology and all the clinical pathology work, which the PCDDP doesn't have in-house. And this then was creating bottlenecks, because you can imagine it's a vaccine situation. So pandemic situation, and you need to be acting fast. You can't then sit and then find the histopathology lab and then you join the queue, etc. So we proposed to have a toxicology hub. So this is a virtual network of labs that has a overarching quality system and way of working. So when a product comes along, everyone is ready to go, and his instruments are validated. The idea is that all the labs would have GLP, so that this becomes a much faster process, right? So in 2023, we had a workshop where we had 34 attendees representing 12 different organizations, and everyone bought into this preclinical GLP toxicology hub project. So that is a project that is currently being funded by the university. It doesn't exist. The concept is still in concept initiative stage, and we're hoping this year to have a follow-up meeting where we can formalize the existence of the hub. So that's where, just to show you how we got here. But of course, the question is, where are we going now? And if we look at the way the world is moving, for me, digitalization is important. So we're looking at also, we have a paper-based system, digitalizing our SOPs and the way we work, having greater translational relevance of our animal models. So I spoke to you that I'm working with the humanized mouse models. That is towards having models that can better predict drug metabolism in humans. So before you even go to the clinical trials, be able to conduct studies in mice that are more representative, that can translate better to the human approaches. Importantly as well, there's something called NAMS, which is New Approach Methodologies. So there's a move away from animal experimentation. Those who are familiar, you would have seen it in the news. And I think because of the familiarity with the DrosAfrica model as well, that would be a new approach methodology. So we are keeping track of this as well. Obviously, if you look in the grand scheme of things, the idea is to minimize research in animals, but one must still ensure that one must validate those replacements. So that means that in our facility and our capacity as an animal facility, we can contribute to validation using animal models, and moving with the times. Eventually, we probably have to look at how do we apply new approach methodologies for toxicology studies, although we're not quite there yet. That links to the new models and technologies. Of course, there's also AI, and we have some ideas. I think there's a lot of work going on in the drug development about using AI to design new drugs and so on and so forth. And I'm sure that they will also be looking at use of AI to predict toxicology. And for me, importantly, is new collaborations. In the Africa map collaboration that I showed you, I'd like to see red everywhere. So I feel that we have a very useful resource for the continent. And looking at capabilities in Africa, I think the reality is that we cannot have necessarily everyone having their own specialized facility. Rather, the model would be centers of excellence in certain areas that all researchers can tap into. So I'm looking forward to have many more researchers from the continent partnering with us for their preclinical work towards developing drugs and vaccines that are relevant for Africa. So thank you very much. That is our website where you can find us. We also have some facility virtual tours, which I will then share with the organizers so they can send it out to everyone. Thank you very much.
Thank you so much for the wonderful presentation. Uh, Hello, can you hear me?
Yes, I can hear you, although um, your screen is frozen, but I can hear you, and I see there's a question that's come through. Can I go ahead and just take the questions?
Yes, please, you can go ahead.
Okay. So I have a question. How important, yes, thank you. How important are partnerships? They are extremely important. As I said, we are not in the business of developing drugs, and we rely on product developers to feed through. We are part of a larger ecosystem, right? So it's important for us to have partnerships in terms of having products that are tested by us, and then that product goes on to the clinical trial partner. So as I mentioned in my presentation, the model that works best for us, we can function as a CRO, like a contract research organization, where people come and we do the work and they pay, and that's fine. But the model that I feel works best, especially in terms of development in Africa, is a consortium model where you have all the partners within the product development pipeline. And then we know that there's something coming through from discovery, and the data that we use will be taken forward to the clinical trial partners. Yeah.
Okay, thank you.
Okay, yes. How do you balance scientific ambition with the realities of funding and infrastructure? That's a very good question. I don't know if you can balance it, to be honest. That is the realities. And as I said, we have a very state-of-the-art facility, and that is the ambition, right? To be world class. We are world class, and it is a challenge to maintain it. It's a challenge to maintain the infrastructure. Sometimes, even if it's too specialized, then you either don't have the expertise within the country, and sometimes you have to bring in people from outside of the country. But I think that if one is aiming for that, and it comes back to the partnerships, if you're aiming that high, then you need smart partnerships that would attract more funding, and obviously you make yourself relevant. So I think that's how one would balance it. What advice would I give to young researchers? If one is interested in it, I would say it's difficult now because I don't know who asked this and in the context of which this was asked, but I would say just pursue. I mean, you can speak to us, for example, set up a meeting with us to understand what is preclinical drug development. Also, in terms of what should you be focusing on, perhaps depending on what level of study you are at, right? Then you would probably need to have a better understanding of pharmacology, and go and read up. I mean, go and read up research papers, see what is being published, keep abreast of what is going on in the space of drug development and vaccine development. And I think that will help you with getting a better understanding of the research area, and also what you would need to do or study to get involved in that. I think what I would do differently is, if I was starting again, if I look at the PCDDP in general, because I joined in the middle, so to speak, I think what I would do differently is probably engage with product developers and clinical trial specialists earlier in the pipeline, to ensure that we have stronger partnerships, so we have a continuous pipeline of products. That's what I would do differently. Okay, so that was the last question. Thank you very much. Once again, it's been a pleasure. I hope that the audience enjoyed hearing about us, and I look forward to receiving emails, and we're open to collaboration. We're open to hosting students as well, joint funding applications. So I really look forward to engaging with the community. Thank you very much.
Thank you so much, Professor. Thank you for taking your time to share your experience and insight with us today. I think one of the most important thing I've taken from here is the capacity to strengthen research. We have platforms like this, which has shown us that there are resources, laboratories, there are infrastructures that have been built in place. And you were asking the other time the context. I think as African researchers, we have so many limitations. And when we see opportunities like this, we want to tap into it. And so we know this is about building collaborations, building people, and we are grateful that we can actually reach out and the lab is open for us to come in. So thank you so much for coming. Unfortunately, we are coming to the end of our session today. And if there are more questions, we will channel them back to you. Also, we want to nudge everyone that we know collaboration is key, research is key to it helps us to grow knowledge and also give back to our communities. It's not just doing the research, we have to bring it back. So collaboration is key, research capacity is key, and that is why the Install Africa is here to create this opportunity. So thank you so much, everyone. This is we're hosting this on the FIRAT platform where we have other events like this. We have the Install Africa, we have the dialogue series. If you miss any part of this discussion, you can go back to our YouTube channel and have a wonderful time listening to our professor give us the breakdown of that wonderful facility there. So once again, we say thank you so much, Professor. Thank you, everyone, our audience that have joined today, and thank you for all the questions, and have a wonderful day.
Thank you.
Africa's biggest discovery science event of 2026, where innovation meets destiny. From microscopes to model organisms, from AI to ethics, we are strengthening the discovery science ecosystem. 8 collaborating institutions, united to catalyze breakthroughs that will transform health, technology, and sustainability across the continent. Discussing building Africa's discovery science infrastructure, model organisms in biomedical research, capacity development for fundamental research, translating discovery science into health innovation, international collaborations for scientific excellence, sustainability and funding strategies, ethics and policy in fundamental research, discovery science in the era of genomics and AI. Scientists, innovators, policy leaders, 150 to 200 global participants, keynotes, posters, workshops, and awards, all in one virtual space. Abstract deadline, September 30, 2026. Symposium dates, December 8 to 9, 2026. Join the movement. Submit your abstract today. Register